I invite Shaziya for the first presentation. Her presentation is going to be about a remarkable topic. That is what I would say. Her title is goes like this: From Transplant Waitlists to Self-Sustained Organs: The Promise of Ectopic Organogenesis. Shazia, you may proceed.
I hope my screen is visible and I'm audible as well.
Yes, it is audible.
Okay. So, good evening, everyone. So, today I'm going to be presenting on the topic which is mentioned here, which goes by from transplant waitlists to self-sustained organs, the promise of ectopic organogenesis. So, starting with a little about me. I am Shazia and I am a final year student at Rajalakshmi Engineering College. I study B.Tech Biotechnology and I have been a scientific content writer at Scioverleaf since February 2024. So let's start on my topic. So first, I'll be telling you about ectopic organogenesis. Now, first, let me tell you about this. The thing is that this is actually a defect in our body. It is a defect phenomenon. So what we'll be actually doing towards the end of the presentation is that we're going to see how we are going to utilize this natural defect to our advantage as a benefit. Okay. So starting off with what actually is ectopic organogenesis. Now, see. This phenomenon refers to the formation of organs or tissues in locations where they're not supposed to be, that is, outside their normal assigned anatomical sites. For example, our heart, okay? The normal anatomical location of the heart is in our chest, which is also called the thorax, right? So when this phenomenon would occur, you'll actually find the heart very interestingly in the abdomen or in our legs, or like it sounds very dangerous, right? So yeah, this can happen, although it is very, very rare. Okay. So yes, and not only you can find these organs in locations that they're not meant to be in, but also they will also adopt a natural functioning capacities in these abnormal locations as well. So you can actually have a beating heart in your legs. Can you imagine that? Yes, it is scary. So yes, as we can understand, this misplacement can also lead to functional impairments and complications. Obviously, it can go from something minor, internal bleeding, to actually something really major, such as permanent deformities or even death. Okay, why? Because these ectopic tissues, like these abnormal tissues, would fail to integrate properly with their surrounding systems, because that is a different location for that organ, right? So it won't be able to integrate with its environment as smoothly as it would in its normal location. So yes, now this phenomenon is observed in humans, right? It is rare, but in what form is it actually visible in humans? So in humans, you will see this ectopic organogenesis mostly in the form of ectopic pregnancy. Okay, so what happens is our the fertilized egg, after that is when a baby is conceived, it is actually supposed to grow in our uterus, right? It is supposed to grow and develop over there. But what will actually happen over here is that our fertilized egg would never reach the uterus. Instead, it will stop in a place called the fallopian tube, which I'll show you later, and our baby would get attached over there and actually grow and develop over there. Now, this can lead to significant maternal morbidity and mortality as well if it is not detected early and managed promptly. Morbidity is like, being scarred with some permanent defect or a dysfunction throughout our life. And mortality is, as we all know, it is death. So it is very important to detect this defect early. Now, why would this happen? Like, are there any risk factors which we should be aware of so that we can prevent this? So these are a few of those risk factors. First is if we have had any prior pelvic surgery. Now, surgeries are dangerous as they are, but if we have an inexperienced surgeon, for example, if he had operated our pelvis for some reason before, and our body is always susceptible to change, right? So if the surgery, even if it had gone as planned, it can actually end up permanently altering the biochemical environment of our organs inside. Okay, so that any small change or disruptance over there can actually also lead to this ectopic pregnancy occurring. And then infections such as chlamydia trachomatis, this is a sexually acquired infection, so even that can lead to this defect. And also lifestyle factors like smoking. We think that smoking just harms the lungs or the heart, but no, it has like far reaching consequences, like it can even affect our reproductive organs, as we can see. Okay, so this is the diagram, so we can understand. So on the left, we have a normal pregnancy, all right? Just explaining the basics of the diagram to those who are new. So on these, the circular parts that we have over here, they are our ovaries, all right? And then we have this fallopian tube as indicated, and then this V-shaped region, which is our uterus. And this narrow pipe down below, it is referred to as the cervix. So what is supposed to happen in normal pregnancy? Yeah, and a very important thing to note is that not many books speak about this, but yes, we often confuse this, that the site of fertilization in humans is actually the uterus. No, it is not. The site of fertilization is actually the fallopian tube, the small tube over the which you can see over there, and actually only the development of the baby occurs in the uterus. So what happens in a normal pregnancy is, in from this bottom narrow pipe like structure, you can see you have a sperm climbing up through the cervix, and then from these ovaries, these circular portions called ovaries, you will have an egg being released. So this sperm actually travels all the way to the fallopian tubes from the reverse direction. They meet the it meets the egg coming from the opposite direction in the fallopian tube, and that is where the process of conceiving happens. The fertilized egg is formed over there, and then it undergoes many divisions, all right? Like, you must have heard of the stages of blastocyst, morula, and it eventually travels to this V-shaped region called uterus, and it gets implanted over there, as you can see in the normal pregnancy. But what happens in the ectopic pregnancy, as you can see, the fertilized egg never reaches our uterus. Instead, it gets stuck over there in the fallopian tube. It gets attached over there and starts dividing, and actually the baby entirely grows over in the fallopian tube. This is not supposed to happen, and this is what the defect of ectopic pregnancy is. So yes, now the general phenomenon of ectopic organogenesis. Now, what are the mechanisms responsible for causing this? Now, there are many processes in our body, which need to be like regulated normally so that everything is smooth in our body, right? Like the processes of cellular differentiation, signaling pathways, environmental factors. For example, we know that like any disturbance, minor disturbance also in these processes can lead to major diseases. For example, cellular differentiation. If this does not happen properly, like cells can abnormally grow forever and ever and even cause cancer. We know that, right? So it is very important for these processes to occur like properly. So any defect, and it can lead to these problems. In ectopic pregnancies, where does the main defect happen? Once again, it is in the fallopian tubes. So what can actually happen? So an important thing to note is that in our fallopian tubes, we have smooth muscles, okay? And there is something called cilia. We must have heard of cilia in even bacteria, right? They are like hair-like structures which facilitate uniform mobility, okay? Like they perform the function of motion. So what do these smooth muscles and cilia do in our fallopian tubes? What they do is they actually, they work together and simultaneously push this fertilized egg into the center of the uterus, where it is actually supposed to implant and grow. But what happens over here is that due to disturbances in the biological processes, we can have abnormalities or even built-in birth defects in our fallopian tubes, such that these smooth muscle contractility and ciliary function do not happen properly, and this can lead to impeding or hindering the transport of this embryo to the uterus, leading to the egg getting stuck over there itself and getting implanted within the tube itself. Okay. Now, research has also been conducted utilizing knockout mouse models. Now, talking a little about knockout, okay? So, and mouse models we know that they are used because of the similarity they have with our body, right? With the human body. So, what does the word knockout mean? So, what happens in knockout models is that there is a specific gene, we deliberately knock it out or inactivate it. and then we use that same mouse model to study our specific disease, because diseases do occur when specific genes are inactivated or mutated or disturbed, right? So what we're going to do in mouse models is that we are going to recreate artificial changes in which we damage the fallopian tube, just like it does in humans when we have ectopic pregnancies, and then we are going to study it. Okay, so this is yeah, so that will indicate that that has actually indicated that there is something called disturbances in endocannabinoid signaling, which also plays a role in influencing this tube transport and embryo implantation. Now endocannabinoids are actually lipid based molecules, all right, and they bind to two receptors. We have CB1 and CB2. CB1 is cannabinoid receptor 1 and cannabinoid receptor 2 is CB2. Okay, so what happens is, like, everything is related to the fallopian tubes, all right? So these endocannabinoid molecules are supposed to smoothly bind to these receptors, CB1 and CB2, which are located in the fallopian tubes again. And these are responsible for actually controlling these smooth muscle and ciliary functions. So a disturbance there can lead to disturbance in these smooth muscle and ciliary functions, and once again, ultimately result in ectopic pregnancy. Okay. So yes, this is this flowchart actually indicates how something such as cigarette smoking can actually lead to can lead to ectopic pregnancy. All right. So what happens is, yeah, so cigarette smoking, the nicotine that is present in it as a core ingredient, that has the capacity to disturb our fallopian tubes at both cell or molecular level and even at the tissue level. In what way? So we're going to see about that. First thing, in cell, okay, so we ingest a cigarette, okay, we use a cigarette, okay, and then first thing that occurs is silation of epithelium. Now, epithelium is the thin lining that lines a fallopian tube, okay. So what happens, silation is the addition of sialic acid, SiA-LiC acid. So what happens is, on our fallopian tube, we have our tube, fallopian tube. On that, we have a thin epithelial tissue. On top of that, we have glycoprotein residues, all right. Now, this silation process, it actually adds sialic acid to the glycoprotein residues which were present on top of the epithelium. Now, what can these do is that this can actually end up altering the biochemical environment of the fallopian tube in a way that it becomes very sticky. The thing is, it is not supposed to be sticky. It is supposed to be only like very fluid, and it's supposed to directly push the egg to the uterus, and it's supposed to only get attached over there. But what happens is, these biochemical changes, they make the fallopian tube only sticky, and our egg gets ends up getting attached there only. And so that is what happens in silation of epithelium. And then we have this ProKR1 and chRNA7 pathway. Now, the ProKR1 pathway, it is actually called prokineticin receptor 1. Okay, and once again, everything is related to the muscle contraction and blood vessel formation. This disturbs that, and the altering this pathway can end up disturbing muscle function in fallopian tube. And the chRNA7 pathway that you can see, this is actually, it stands for nicotinic acetylcholine receptor alpha 7. Okay, now this is the exact pathway that gets directly affected by the nicotine that we ingest when we are smoking. Okay, this actually ends up disturbing those ciliary particles, those hair particles in our fallopian tube. Okay, and then we have the E2ESR pathway, also estrogen estradiol pathway, which also gets affected. One second. Yeah. And so these were the disturbances that can occur at cell or molecular level. Now, what can happen at tissue level? So at tissue level, what can happen is that obviously the muscle contraction process can get disturbed, the ciliary swaying, the hair, the ciliary hair, they're actually supposed to beat uniformly so that the egg gets pushed in a uniform motion. But even that process is disturbed, once again causing our egg to get stuck right in the tube. And then membrane secretion, surface adhesiveness, surface adhesiveness increases, it is a problem because our egg would get attached there only. So all of these problems can be caused by cigarette smoking, ultimately leading to ectopic pregnancy. Okay. So yeah, so once we are diagnosed with ectopic pregnancy, what can it result in? So it can result in life-threatening complications such as multiple internal organ bleeding, and we need to detect it immediately using a medical intervention, or else it can be dangerous. And then so in women who have already experienced one case, the risk of a second ectopic pregnancy is also increased by default. Okay, and also rather than just ectopic pregnancies, there have also been reports of ectopic thyroid tissue. the butterfly shaped gland in our neck, it can actually be it can actually grow in places which are not the neck, okay, in any other portion of the body as well. So when this happens, it is actually very difficult to diagnostically use the existing diagnostic methods that are present in medicine. It is only developed in a way that we can use it for where the organs are normally placed. So when the organs show up like this in a totally different route, it is very difficult to surgically or medically intervene. So it is a challenge for the doctors to treat this. So yeah, research is being conducted for crucially as it is crucial for improving diagnosis and treatment strategies. Okay, so now we saw about the defect the ectopic organogenesis can be, right? Now we'll also see about how this defect is being utilized as an advantage. So what recent studies have been done, especially by a doctor called Eric Lagasse, we'll be talking about it later. What he has shown is that we can use healthy liver cells or hepatocytes, which are the functional unit of liver. We can actually transplant this into lymph nodes where they not only grow and survive, but they also function as mini livers. You can actually grow mini livers in our body in places which are not the liver. Like, that is cool, right? So so one important difference to be noted is that this is not directly an organ transplant, because the liver cells are not being directly transplanted to the liver. Instead, we are growing it in a different environment called the lymph nodes. We a little to know about lymph nodes is that they constitute the primary immune system. Okay, we have our spleen and thymus. They constitute the primary and the lymphatic fluid, which constitutes the primary immune system. Okay. Yeah, so this approach suggests that lymph nodes could actually serve as bioreactors in our body itself. We have bioreactor machines which can help us in growing organs, and they are a novel avenue for treating end-stage liver disease and potentially many other organ failures, different types of organ failures. So we'll just see a little about this from from a research paper titled "Ex vivo cell therapy by ectopic hepatocyte transplantation treats the porcine tyrosinemia model of acute liver failure." This is what we are doing over here is that we're actually treating acute liver failure in pigs affected with tyrosinemia by this method. Okay. But the only thing is that we are not growing the liver cells over here in the pig's body itself. We're growing it in a different body, and then we're using it to treat the pig's liver failure. Okay. Yeah, so this is how the process is done. Initially, the hepatocytes are transplanted directly into the lymph nodes of the organism. The pig over here, the pig is concerned over here, and the lymph nodes, they're very rich in immune cells because they constitute the primary immune system, right? And they have a unique microenvironment which is conducive to cellular organization and function. Now, the research led by Dr. Eric Lagasse, it has demonstrated that these transplanted hepatocytes can not only thrive in the lymph node environment, but they also form structures that mimics the functionality of a mini liver. Now, why is a lymph node such a good environment for growing these organs? That is due to three factors. Okay, mainly immune modulation. Now, since the lymph node is actually already a primary immune system location, we have many cells located in these lymph nodes that prevent rejection, which is a very common problem in organ transplantation. So yes, so since the lymph node is naturally equipped with so many immune cells and immune networks, like the chances of rejection get reduced by 90%. Okay, and then vascularization. Not only immune cells, our lymph nodes also possess a very rich inherent vascular network, that is a network of blood vessels, okay, that supplies this artificial organ with all of the necessary blood supply needed for cell survival and function and growth. Okay, and then we have cellular communication. Okay, so yes, not only and it is important to note that this lymph node is not an isolated location. It actually can communicate with the entire body. So yes, we can actually establish it already in the body that we have new transplanted hepatocytes, and the surrounding tissue also responds and coordinates with it. So like overall, the body will be readily accepting it rather than rejecting it. So yes. So in this case, what was done is the transplantation of autologous hepatocytes was done. Autologous means derived from the body of the same individual in which we're treating the disease. Okay, so the transplantation of autologous hepatocytes was done into mesenteric lymph nodes. Mesentery is the region between intestine and the stomach. It is actually a fold of tissue that attaches both the stomach and the intestine. Okay, so and in that fold of tissue we have lymph nodes. So the transplantation of these liver cells was done in that location. Okay, and this pig was suffering from induced acute liver failure. So yes, and these lymph nodes were the bioreactor for the hepatocyte survival and function. And then after doing this, it was actually found that the pigs exhibited significant recovery in liver function after the completion of the whole process, that is. And not only did the hepatocytes survive, but they also organized into like structure that exactly resembles the normal liver, complete with the bile ducts that is located adjacent to the liver and vascular networks. So as research went on, this was like, where like increasingly adopted by the scientific community as a viable therapeutic strategy because it showed success in a large animal model which holds even more similarity to us. So yes, this was a small overview, pictorial representation of the research. So the first diagram, it actually indicates that liver was cut, the cells were isolated, they were transplanted into the intestine and stomach mesentery regions, lymph nodes, and then the growth occurs. And this is the liver structure that was actually obtained from that mesentery region. Okay, after the growth, the pig was euthanized, cut, and then the liver tissue was obtained. And then this comparison was done using an MRI. In the right, we have normal liver tissue, and then on the left, we have the liver tissue that was growing in these mesenteric lymph nodes. They were actually found to have a lot of similarity, indicating that successful liver growth was done. So yeah. Now, why do we need these? Okay, like why are we addressing using this as a therapeutic strategy? Yes, so that is because the number of people needing transplants increases every day. Like kidney transplants are the most needed, and liver comes second, and lung comes third. And 3 to 5 years is the normal waiting time for a kidney, for a kidney transplant awaiting person. They actually have to wait for 3 to 5 years before they get the perfect donor. It is very difficult, and so many people end up getting neglected of the medical help needed at the right time. And then we have 84% patients currently waiting in need of a kidney. And liver damage also, liver damage starts with fatty liver, and then liver fibrosis occurs. There's growth of fibrotic tissue, and then we have liver cirrhosis, in which the organ ultimately dies. And it is very difficult to find a liver donor also whose bone marrow completely matches with us. So that is why, and the last option for survival is also liver transplant. That is why it's so necessary that this method increasingly gets adopted as a successful treatment method. Yeah. So we'll just yes. So this is another method in which we are using a mouse model. All right. This is first what we're doing over here is we are using a blue dye. We are injecting it into the mouse's lymphatic vessels. This blue dye actually increases our visibility, and then over there we are injecting hepatocytes actually using a 28G needle insulin syringe. And then we're observing using MRI, we're observing the results, and we see there is a lot of similarity to the normal liver and the apparent liver that is growing in our mouse's lymph nodes. And yes, so this is like the result of the process. Okay, so jejunal, axillary, and popliteal. This actually, jejunal is the region in our near our intestine, axillary is the armpit region, and popliteal is the region behind our knee. Okay, so the first rows 3 pictures, they indicate the normal tissue, and the second rows pictures indicate indicate the liver growing in those regions. Actually, you can see the difference. For example, in the jejunal region, you actually do not have any tissue there, and in this bottom picture, you actually see a full-grown liver tissue over there. The thing is, the liver has been successfully able to grow in all of these 3 regions, which is actually a remarkable success. So let me just quickly conclude with the future therapeutic scope. As we already spoke about, that using these lymph nodes as bioreactors, we were able to generate solutions for mainly till now only end-stage liver disease. But this also, it is a potential solution for multiple organ failures such as the pancreas, kidneys, thymus, and to generate functional tissues in these organs, addressing multiple areas of medical need. And yeah, so this is a small example of growing an ectopic thymus, and also an example of growing what do we say, ectopic pancreas for treating insulin and bone marrow issues respectively. So yeah, that is all about it. Thank you so much for listening, guys. If you have any questions, I'm ready to answer. Thank you so much.